How many of you know what your blood type is? Well, the reason you have this information is because of this man, Karl Landsteiner. In this presentation, I will be going over Karl's life, the research he did, and how he was able to do it. Karl was born June 16, 1868, to Leopold and Fanny Landsteiner. Karl was born an only child to a Jewish family. Leopold had a doctorate of law and was an esteemed journalist and publisher in their community. Unfortunately, Leopold died when Karl was only 6 years old, leaving him in the care of his mother, Fanny. Because of the lack of a father figure, Karl became very, very close with his mother, Fanny, and over the years, they continued to establish that relationship. And when Karl was 40, when his mother died, he had a death mask made in her remembrance, and it hung on his bedroom wall for the remainder of his life. Despite their close relationship, Fanny wanted her son to have a better life, and so she sent him off to school. Karl began state school in Linz, Austria at the age of 12. Upon completion of state school, he moved on to medical school in Vienna, Austria. Early on in his educational career, Karl expressed an interest in hematology, and one of his first published studies was done while he was a medical student and discussed the effects that diet can have on our blood composition. Karl also found interest in organic chemistry, and upon graduation of medical school at age 23, he chose to pursue postdoctoral research in organic chemistry instead of practicing medicine. Karl would spend the next 5 years in Zurich, Switzerland, studying under Emil Fischer, a Nobel Prize-winning chemist. After spending several years studying organic chemistry, Karl decided that he wanted to return to medicine, and so he returned to Vienna and applied for a position as an assistant at the Hygiene Institute of Vienna. At the Hygiene Institute, Karl was able to re-spark his interest in immunology and antibodies. A year later, Karl was transferred to the Department of Pathological Anatomy, where he continued to study in immunology, but also began to study morbid physiology. It was here that Karl was able to publish over 50 studies that ranged from topics from syphilis to polio and to haptens. One of the first research projects Karl took place in at the Department of Pathological Anatomy in Vienna was a study on syphilis. Karl was the first scientist that was effectively able to transfer syphilis from human subjects to monkeys so they could further the research on syphilis and to find a cure. Karl also spent significant time researching polio, and he was the first scientist to find that polio came from a virus. Karl's arguably most important research was actually done on haptens, which is a small molecule that's found naturally in the body that poses no threat to our immune system. However, when these haptens are joined with a larger carrier, such as a protein, they often elicit an immunological response. Karl was able to make this discovery of these haptens in the body, and he was able to pioneer synthetically engineering these haptens for further immunological research. Karl argued that one of his most important research and significant findings was his research in haptens. However, that's not what he's known for. Karl is known for his studies and research in hematology. Before Karl, scientists knew that blood transfused from one animal to another animal would elicit severe responses. However, the complexities of transfusions between one human to another human were still unknown. Karl was the first scientist to propose that when transferring blood from one human to another was causing shock and jaundice, these responses that had been seen in several transfusions before. After proposing this hypothesis, Karl began his significant research into blood. He spent several years looking at the different interactions blood have with each other, and he would take blood from one individual and mix it with the blood from another individual. And what he found most often occurred is that the blood would clump with each other, which is also known as coagulation. Other scientists believed that this phenomenon, this coagulation, occurred because the blood had an underlying disease within the blood. However, Karl's research found that even healthy blood can coagulate. So after several years and several research experiments, he found that the reason that the blood coagulates is because the blood has different types of antigens. And Karl proposed that there was 3 blood types, A, B, and C. We now know them as A, B, and O because they were renamed after he found the 4th group, AB. And as you can see from this chart, we have the 4 blood groups, where blood type O doesn't have any antigens, where blood type A has an A antigen, blood type B has the B antigen, and blood type AB has AB antigens. And the reason that these bloods, when mixed, cause coagulation is because they have antibodies for the different types of antigens. So it's very important that you have the right blood type when mixing bloods. Karl also had significant research in the Rh factor. He was the first scientist to identify that there was another type of antigen present on red blood cells that did not correlate with the ABO system. Rh factor poses a significant threat to pregnant women because if a Rh negative mother is pregnant with an Rh positive baby, her body will often start to produce antibodies to fight the new antigen. With the understanding and knowledge that we got from the Rh factor from Karl's research, we now have been able to prevent mothers from preventing antigens or antibodies to fight the antigens of their unborn babies. Karl's research in hematology and the understanding that we have of the ABO system and Rh factor has made a significant difference in our medical field. 4.5 million Americans need blood transfusions each year. And because of Karl's research, we can safely and effectively transfuse blood. Karl received many awards and nominations for his research in hematology. And as you can see, he won a Nobel Prize in 1930. He also received the Dutch Red Cross Medal and became an elected honorary member of the Royal Society. In order to receive all of these honors and awards, Karl spent most of his time in the lab. However, he did make some time for his personal life and family. In 1960, Karl married the love of his life, Leopoldine, and a year later they had their son, their only son, Ernest Karl. Later in his life, Ernest would follow in his father's footsteps and become a surgeon. In 1937, Karl moved his family all the way over to New York in hopes to give them a better life, where Karl became a member of the Rockefeller Institute. Karl in his free time liked to play piano or read detective novels. And similarly to the novels he loved and enjoyed, his life ended in somewhat of a tragic manner. A few years after moving to New York, his wife, Leopoldine, was diagnosed with thyroid cancer. And when he was unready to let her go, he decided to start researching into a cure. Unfortunately, Karl was never able to find a cure for thyroid cancer, as he had a heart attack in his laboratory at age 75, and 2 days later, he died of heart failure. His wife died a few months after him. As you can see, Karl Landsteiner's research into hematology and immunology has made a significant difference in our medical society and medical community that we have today. Without Karl's research, we wouldn't be able to safely transfuse blood that saves millions of Americans each year. Karl also made significant differences in diseases and transmitted diseases such as syphilis and polio. These are my works cited. Thank you.